Okay, so I am just going to try to do some of my weekly announcements in a video format, and that way, then hopefully that will help you to kind of see what areas of Canvas that I really want you visiting for that week. I know some of you are kind of feeling bogged down with the Canvas site. So, and I actually did make a few updates. I realized when I course copied over the course from last semester, the lab was a hybrid format. And obviously, this is an all online format. So there's extra resources that I put in there for you. So what I did was I went ahead and added back in some of those resources for those of you that might have felt a little lost on the histology. So hang in there with me, and we'll get all of these initial beginning of the semester kinks worked out. But I'll go ahead and make sure that I update all those labs for you, so that way then you have some of those extra resources. And hopefully, kind of by doing this week to week announcement on Sundays, you guys will kind of be able to get started off on the right foot, and then I can catch any kinks or things that didn't make it into the Canvas course that I thought were initially in there. So hopefully that will help us both. So let's go ahead and dive in right now into what you guys are supposed to be working on for this week. So if you go onto the home page here, let's talk about what you're going to be doing for lecture. And again, remember that you can always go back to the syllabus if you want to just see what's due really quickly. So you can scroll down here, and that way then you'll be able to see, all right, what is going to be due this week. So, next, basically don't have anything due until September 2nd, which is that chemistry quiz. And then on Sunday, you're going to have the week 2 muddiest point. So, but again, just because you don't have anything due during the week doesn't mean that you shouldn't be chugging away at the lecture and the lab. So let's talk about that. So let's go ahead and get into what you're going to be doing for lecture this week. So again, you're going to be going to exam 1, which is going to be all of your lectures for this first exam. Hopefully you've already gone through the introduction and you're on to chemistry. And hopefully you had a chance to do the first part of the chemistry lecture last week, and you're going to dive into the second part of the chemistry lecture this week. So you want to go ahead and finish that in the beginning of the week. Now, this hopefully should be a review for many of you. If you just had Bio 181, you're like, oh, this is easy peasy, right? But for some of you, and so if you're in that, this is easy peasy, please watch the fast track lectures that I did here. I didn't go quite into as much detail. I do teach this course for a fast track 8-week course, which we kind of expect that you have recently had a lot of this information from Bio 181. But again, I know that some of you are in that category. It's been a few years since you've had this information. So take the time to watch the 3-part lecture on the chemistry series that I would do for sometimes my 16-week courses. And that way then hopefully it will give you a little bit more information and maybe kind of help you really jog your memory on some of those concepts that you haven't seen for a while. So again, these first really 3 lectures should be a lot of review. And if they're not, you may have to take a little bit more time here initially in the beginning than you maybe had initially planned for. But it will be worth it in the end, because believe it or not, all of this stuff that you have in the beginning here is very important. So, in the chemistry lecture, understanding ions becomes vastly important when you talk about things like the urinary system, and you talk about some of these ionic imbalances. So that can occur if things aren't going right. So again, really important that you kind of understand what an ion is before we get into that in the systems. So, and again, you can see here, So again, you can either watch the part 1 and part 2. Hopefully, again, you already watched part 1 last week, but you're on part 2 of the fast track, or you can watch. You don't have to watch all of these. This is or. If you want to watch the longer lecture series, you can watch part 1, part 2, and then part 3. Okay. And then you'll see the chemistry quizzes here for lecture. So when you're ready to take the chemistry quiz, again, you can take that anytime before 11:59 p.m. on September 2nd. And again, you have about 15 minutes to finish once you start. Okay? So that's the first half of lecture this week. So then you're going to go ahead and click back to exam 1. And then you're going to start for the second half of the week, you're going to start into the cell structure and function lecture. And so again, some of you have told me that you like The Office. And so I try to stick in some memes in there every once in a while. So I couldn't resist this one. But anyway, so as you go through that, you'll see that here is the PowerPoint for the cell lecture. Sometimes I'll put the presentation, if it's sometimes a bigger file, I'll put it in a PDF format. And then I also have been doing it as an outline, sometimes because last semester I had some students that requested the outline. So again, you only need to do one of these. If you like the PowerPoint, you can do that. If you just want the smaller file in the PDF format, you're welcome to do that. But you don't have to look at all of these. Just pick which format you like. And then you're going to go ahead and watch the lectures. I did these, there's 5 parts. I did them in shorter clips. So you can that's why there's so many parts to this. But yeah, I would say for this week, if you can get through to part 3, that would be great. And that way then you'll really be kind of keeping up with things, and then save, part 4 and 5 for next week. Or if you can want to cruise ahead a little bit, go ahead and cruise ahead and get through all of the cells, especially if it's a review for you. And then you'll be a little bit ahead of the game. And then again, this quiz isn't due until September 9th, since, again, we're finishing up cells next week. Okay? So that's kind of where we're at in terms of where you should be spending your time in lecture this week. And then I do have an idea for some of you. So I know many of you have sent me the time management Excel sheets, and I'm finding that many of you have very, very full schedules. So it would be nice if we could start a discussion board, and you guys, if you wanted to create like some Quizlets, and maybe you could talk to each other and say, hey, look, I'll create a Quizlet for, this first chapter, the introduction, and then, somebody else, you do, the second chapter, a Quizlet for that one, and then, the third one. But I would love for the discussion board to be a place where you can share resources. Anytime that you guys, because honestly, with anatomy and physiology, repetition is key. The more that you can go over this information, the more it's going to stick in your brain, right? But it becomes very challenging if you have a busy schedule. And so if you guys can share study tools, that would be the ideal. So that's my big suggestion for you all. And if we can I'll maybe just create a discussion board thread for that. And then you guys could just all, hop on and put in whatever resources you have that you create. So that's kind of my suggestion. Also, I would like, if you guys, for the muddiest point, so again, we also have the muddiest point due this week. Again, you can go back to the syllabus and then see you've got, okay, week 2 muddiest point is also due on Sunday. So I would love for the muddiest points for each week to be either a place where you can ask questions that you're like, you're not kind of familiar with a certain concept, and you're like, I didn't really understand this. It was really my muddiest point this week. Or, if you didn't really have a muddiest point, but maybe you're just having trouble memorizing things, maybe you guys can each give like one mnemonic that you came up with for a concept that was tough for you, or some type of studying tool that you used to help you get that information. So I would love for this muddiest point to be a place where you guys can share information. as well as ask questions. So it doesn't have to be just asking, questions. Okay, so let's now get into the lab, because this will probably be a little bit different for you in terms of, and you guys will probably be much happier with this for the histology, because I didn't realize that I did not have my PowerPoints, and I created for histology. I created some PowerPoints for my in-person students because I felt like it was so hard. It's so difficult to tell what you're looking at. And so here you can click on epithelial tissue. So now that when you start the histology labs, for each of the histology labs, and so let me actually go back here. So you're going to see for these histology labs, it's going to be composed. You're going to have 4 different tissue types that you're going to be focusing on. And these are what we call the general tissue types. So you're going to have epithelial tissue and connective tissue this week. And then you'll have muscle tissue and nervous tissue next week. We combined muscle and nervous tissue because there aren't as many slides for each one of those as there are the epithelial and connective. So that's why that's separated out that way. But for each of these 4 general tissue types, okay, you are going to ask yourself these 4 questions. What is the general tissue type that I'm looking at? So again, by general tissue type, we mean, is it epithelial tissue? Is it connective tissue? Is it muscle tissue? Or is it nervous tissue? Okay. Then we're going to get into the specific tissue types, and I'll talk about this in just a minute, but you can see with epithelial tissue, here are all of the specific tissue types. So then, is it simple squamous epithelium? So these are all, again, the general tissue type here is epithelial tissue. And then now we're going to get into these specific tissue types. How do we break up epithelial tissue? So we break up epithelial tissue into all of these different types. There's more epithelial tissue even than this. We just have, a few of them. But so there's simple squamous epithelial tissue. There's simple cuboidal epithelial tissue, simple columnar epithelial tissue, non-keratinized stratified squamous epithelium. So, excuse me. There's all of these different specific types of epithelial tissue. So when you get to the practical or even the practice quiz, you're going to have to be very careful to pay attention to how the question is being asked. So we could, 1, ask, what is a general tissue type? So let's say I had a slide of, I'm just saying this for, general sake to just kind of give you an idea of simple cuboidal epithelium. Okay. So if I ask you, 1, what is the general tissue type? It's epithelial tissue, because remember there's only those 4 general types. If I ask you what is the specific tissue type, then you would say simple cuboidal epithelium. Okay? Then the third question I can ask is, give an example of a location of where you would find it in the body. And I said, try to learn like at least 1 or 2 examples so that you it will, to be honest, it will help you for not only 201 but 202 if you learn that, because you're gonna see all these tissue types again. They're gonna pop up when we go into these different systems. So for example, simple cuboidal epithelium here, 1 location is in the kidney tubules. And so when you get into 202 and we talk about the kidneys, then you're gonna go back and kind of remember the simple cuboidal epithelium, because it's going to be lining your kidney tubules. So again, it's just a good thing to, have a couple, and I kind of put some of the main locations where you find these. But on the practical, again, we'll just say, I might just have a picture of the simple cuboidal epithelium, and I'll just ask you, give an example of where you would find this in the body. Okay. And then the 4th one is going to be, what cellular characteristics is this pointing to? So you're going to see each of these types of epithelial tissue have certain characteristics that you have to be able to identify within the slide. So where's the lumen, where's the apical surface, the nucleus, and the basement membrane? And you may be like, well, where in the heck am I supposed to get all this information? Like, am I just supposed to pull this out of my head? I don't have this information. And so this is where, and I apologize that it was not linked before, my PowerPoint is going to help you out. So this is basically like the lab book introduction to histology in a PowerPoint. So I'm going to go ahead and pull this up for you so you can see this. And so you'll see, I remind you, I tell you what a tissue is here. And then we talk about the 4 general tissue types that we talked about, remember? So epithelial, connective, muscle, and nervous. And then the 4 main questions that I will ask you on the practical. And then we get into, let's talk about epithelial tissue, right? So then I say, okay, well, there's all these different types of epithelial tissue, and these are different places that you can find them, okay? So again, for example, stratified squamous epithelium, especially if it's non-keratinized stratified squamous epithelium, you can find in the esophagus. Simple squamous tissue you can find in the alveoli of the lungs, and so on. Simple columnar you're going to find lining the gut, and so on. So again, you can kind of go through all of these slides and talk yourself or walk yourself through them, but it's a really good introduction, and you'll get this in lecture too. So I don't want to, it's so hard because I don't want to make you listen to this twice. You are going to get this in lecture, and you may find actually that if you can work ahead a little bit and get into the epithelial lecture, that will help you with the lab. But again, because you're getting the lab prior to lecture, I think that hopefully this PowerPoint will really help you out. So here you can see we talk about epithelial tissue, and that with epithelial tissue, you're always going to have an apical surface and then a basal surface. So let me show you what that is. So the apical surface is going to be what's called your free surface, and it's always going to be kind of exposed to either the outside. So if it was on your skin, right, the apical surface of your cells would be exposed to the outside world. Or if it's, lining some organ, and it would be basically the apical surface would be open to the cavity, the body cavity. So again, what we call a lumen. So for example, if you have lining your intestines, you have what's called simple columnar epithelial tissue. And they actually kind of look like this because they look like big columns. And so lining the intestines, you would have these cells. And here inside, here would be the lumen. So this is the apical surface, and this would be the lumen, and this would be where the food chyme is moving through. So this would be where the food's moving. And so that apical surface would be exposed to that internal cavity, okay? So hopefully that'll help you out a little bit. And then the basal surface is always going to be kind of connected to kind of what we call a basement membrane below, and then to connective tissue connecting it to whatever's below, okay? So hopefully that will help you out a little bit in terms of identifying things. And then when you look at the classification of epithelium, you're going to see that they use these, and we use these different terms kind of regularly with epithelial tissue. So one will use the term simple or stratified. Simple means it's just one layer, okay? You just have one layer of cells, just like you have here. If it's stratified, you have multiple layers, okay? And then you'll see I'll talk about pseudostratified. You do have an example of pseudostratified ciliated columnar epithelium. And this one's goofy because So look at this. It looks like it's multiple cell layers, but it's not actually, because they're actually all attached, but they're just all squished in here. And the nucleus are in like different levels of the cell. So like here, the nucleus is at the bottom. Here, it's in the middle. So it makes it look like it's a couple cell layers thick, but it's actually only one cell layer thick. So what we do is we call this pseudo-stratified. It looks stratified, but it's not. So pseudo means false, right? So it's like falsely stratified. And then ciliated columnar epithelium. So they're columnar cells, which we're going to talk about in just a minute. And then you can see that, and I'll give you hints on these, because a lot of times people confuse pseudostratified ciliated columnar epithelium with stratified squamous epithelium. And the big difference that you can kind of identify is in terms of these two. This one looks like it's like 20 cell layers thick. Like, look at that. That's a lot of cell layers thick. And then the pseudostratified, that's really only one cell layer thick, but it usually looks like it's like 2 to 3 cell layers thick. So that's how you can tell them apart. This looks like it's like 2 to 3 cell layers thick, even though it's only one. And this one looks like it's like 20, okay? So you can see that it looks pretty different. So you can look at all those crazy cell layers squished in there, okay? So anyway, let me go back here. Again, I'm just kind of introducing these terms. So you'll name it based on your arrangement of cell layers, right? So either it's simple, one cell layer, or stratified, many cell layers, or again, we have that extra pseudo-stratified, which is really simple, but it makes it look like it's multiple layers. And then we have the cell shape. So we have squamous cells, which are going to be these flat pancake-like cells. We have cuboidal cells, which is just like it sounds. They look kind of cube-shaped. And then we have columnar cells, which are going to be obviously column-shaped, okay? So you're going to see they're all named after that. Like here, this is simple squamous epithelium. They're simple flat cells, okay? And again, I'm going to go over all of this in the lecture, but you can see they're, because they're just one single layer, they're very flat, they're going to be in places where we want to do diffusion, okay, across the membrane, and things need to readily diffuse across the membrane. That's why I always tell people, if you know that, and you think about the functionality of the cell type, then you'd be like, oh, okay, that makes sense that would be in the alveoli of the lungs, or you can just put lungs. As long as you put lungs for where you would find this, I'm happy. Because then I know that you're like, okay, this is only a single layer, one cell layer thick. That's perfect for, diffusing oxygen and carbon dioxide across the membranes, right? In the lungs, so that we can bring oxygen in and get rid of the carbon dioxide. So we need these thin, flat cells and only one layer, so we can do that quick diffusion of those gases. Okay, so again, those are little tricks to kind of help you remember those types of things. And then this slide might be a little confusing, just because you're like, whoa, there's a lot of different cell layers here. What you're focusing on here for the simple squamous epithelium is just this outer layer. That's called actually the serosa. You'll learn about that when you get to the digestive system. We're not obviously close to that yet. But here, this kind of outer layer here is simple squamous epithelium. So, and then you can zoom in, and now you can really see that this is simple squamous epithelium, okay? So, sorry, I didn't mean for this to be going through each of these slides, because I know you all can read, but and this is simple cuboidal. Great place to find it is the kidney tubules. Here, you can look at these. We're going to have these kind of different sections for the tubules. So you'll see we have longitudinal sections where it kind of looks like you cut through, like you took if you have a hose, if you have a hose, right? And you cut longitudinally through it, and then you're looking into, you like cut the hose in half and you're looking into it. That's what you're seeing here. And you have these simple cuboidal epithelial cells that are lining that. And then the lumen inside. Or you can you can cut it here, like I just chopped it here, and now you're looking into, that's the cross section, you're looking into the tubule, okay? That way. So that hopefully helps you out in terms of sections on what you're looking at. And so you can see again here you have, this is a longitudinal section, and you have these simple squamous epithelial, or I'm sorry, simple cuboidal, not squamous, simple cuboidal epithelial cells. And you can see they're cube shaped. It's kind of nice. And they're lining again, they're lining the hose, okay? And then in the inside of the hose is the lumen here. And so, and the apical surface is going to be the surface of those cells that's basically facing the lumen, okay? So, again, you can kind of look at those, and here's a cross section. So again, it's like you're looking into the hose this way, right? So you cut it and you're looking into the hose this way. So you can see again these simple cuboidal cells, and then you're looking at the lumen. That's going to be apical surface. This will be your basal surface and things like that. Okay. So then you can look at simple columnar epithelial tissue. The big place that you're going to find this is in the GI tract. Okay, simple columnar is GI tract. So here you see this same cross section of the intestines, but now here you're looking at all this folded because the intestines again are gonna show up, especially in a section. They're gonna kind of show up folded. Not gonna be this beautiful, perfect, round circle. So here's the lumen. Here's the inside of the intestines where the this clear part where the food would move through, and then here are the cells lining that are going to be right here. Those are your simple columnar epithelial tissue. Okay, and the nice part is this is going to show you some really important things that you're going to see. So lining our intestines, we need to have mucus that we secrete on our intestinal lining to help the food kind of move through there so it doesn't damage these cells. So that mucus kind of helps kind of lubricate the inside of the intestines. And the cells that produce that mucus are called goblet cells. So they'll release that lovely mucus. Okay. So again, and then you need to know the apical surface and the basal surface. Again, I'm sorry I'm going more into this than I really had wanted to, but I just feel like as I'm getting into it, there's a lot that you guys haven't had in terms of lecture yet for this. So again, if you want to watch the lecture prior to getting into this, that is also a great idea. Pseudostratified ciliated columnar epithelium, again, is going to look pseudostratified, but again, it's only one cell layer. It just looks like it's 2 to 3. And then it has cilia. It's ciliated, so it has these cilia on the surface. And so you're going to find this in things like the respiratory tract. And so right now, excuse me, and as I'm coughing, I have a respiratory infection, and so just a viral one. And so again, I'm producing a little extra mucus, right? And so what happens is that mucus that traps bacteria and viruses that you're breathing in and dirt that lines the airway. So this lines your airway. And as you secrete that mucus, then you don't want that mucus with the bacteria and stuff to settle down in the lungs. You want to bring it back up to the back of the throat so you can swallow it. So what happens is all these little cilia actually push that mucus and all of that lovely bacteria and stuff that it traps in that mucus up your respiratory tract and back into the back of your throat, so then you can yummy swallow it and then bring it down into the stomach, right? Because the stomach is a much better place with its hydrochloric acid in there for all that bacteria and the viruses, okay? And then your lungs. So you'll see that. So here you're going to zoom in, and you'll have to look. There's going to be different cell layers here because we use these slides for multiple tissue types. So you're going to focus in on the ciliated pseudostratified columnar epithelium, just this right here, okay? And then you can zoom in So, and then this is the lumen of the airway. So you're looking at stuff that's lining the lumen. And that's where a lot of your epithelial tissue is lining. It's lining the outside and then the inside of these tubes that go through the body, like the respiratory, right? The respiratory tract has kind of a tube that goes in the body. The GI tract has a tube that goes all the way through, right? So that's where you're going to find a lot of these epithelial tissues. So you can zoom in and see here's that ciliated pseudostratified columnar epithelium. And then you can see here all of those little cilia here. You're going to see, again, goblet cells that are producing that mucus. Yum, right? And then here's the cilia that are going to push that mucus and all the things that it traps in it to the back of your throat. Yummy, right? Stratified squamous epithelium. So you're going to find this. You're going to see it's really thick, right? So really thick. Many cell layers. You're talking about like 15 to 20 cell layers. So it's a lot of cell layers crammed in there. And so you're going to see, you're going to find this lining things like the skin and the mouth and the vagina. These are going to be areas where you need some extra cell layers. Okay. And then you'll also find it in like the esophagus as well. So here you can see this is a mouse esophagus. And so here, inside here, this, again, here's the lumen, okay? And so, and the esophagus, here's the esophagus. It's like a goofy, doesn't look like a pretty round esophagus, right? It's kind of folded because this was taken from a dead mouse. So here is the esophagus, and you can zoom in and see, look at all those cell layers. I mean, it looks like it's probably about, maybe 15 cell layers thick. So a lot of cell layers. So that's how you know it's stratified squamous. And then I'll show you how to tell this is non-keratinized, meaning we don't have the keratinized layer like we do on our keratinized waterproof layer on the outside of our skin. And I'll show you what the keratinized looks like in just a minute. So here again is that non-keratinized stratified squamous epithelium. And of course it has the free apical surface facing the lumen. And then there's the basal surface up against the basement membrane. This is going to be your keratinized stratified squamous epithelium. So you can see this keratinized layer here of all of these kind of outer, sorry, I just hit that slide there. All of these outer dead cells that you'll see again. And then there's deposits of keratin in there as well. And that's what gives us that nice tough surface on our skin. So you're going to find this in your skin, the keratinized stratified squamous epithelium. And you'll see, again, it's pretty obvious as you zoom in here, you can see this keratinized layer right here. Okay. And so all the way from here on up to here is the stratified squamous epithelium. Okay. Below this is going to be some of your connective tissue in the dermis. So all of this right here is going to be that keratinized stratified squamous epithelium. Okay, and you can kind of zoom in on that and see that. And then there's transitional epithelium. I give you the hint that the way that you can identify this from like the stratified squamous is that this looks to be about 5 cell layers thick. So if you look, like it looks about 1, 2, 3, 4, 5, maybe 6 cell layers thick. So if it looks like it's a multi-layer tissue and it's like 5 to 7 cell layers thick, then it's probably transitional epithelium. Again, here you're looking at this, you're like, this is like 20 layers thick. And then obvious, this one's easy because it's keratinized. So then that it's stratified squamous. But here, this one, you might be like, I'm not sure which one this is. So this one you can look and see, it looks like it's probably about 15 to 20 cell layers. You got a lot of cell layers here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So again, then that that's not transitional, okay? So transitional is going to look like it's about 5 to 7 cell layers thick. Also, a lot of times you'll have kind of some more bubbly cells, although you can't see it here. You'll look here, and these cells, they can kind of be stretched. So here they're kind of a little more stretched, or they can look here when it's relaxed, it looks kind of a little more bubbly, right? And so that's because this is going to line the urinary tract, ureters, bladder, and urethra. So this is a tissue that needs to be able to stretch, right? And so it looks like this when it's more stretched. So here you can see this is the lumen of what's called the ureters. So the ureters actually are going to go from the kidneys. They're the tubes that go from the kidneys down to the bladder. And you can see here's the lumen. I know you might not know that yet, but that's why I kind of explain it so you can see. But it is part of the urinary tract there. And so you can see here's the lumen, and here's the transitional epithelial layer. And then here you can see it forms these nice bubbly cells. So looking good. I think, so that's the end of your epithelial tissue stuff. And sorry, again, I didn't mean to go into all this. I think I'm actually just going to do a separate video for connective tissue for you guys because you are wanting, I do want you to get into connective tissue this week. But what you're going to do, so with these, and I'm so sorry this has ended up being so long, but what you're going to do for lab is you're first going to download that PowerPoint, go through that PowerPoint. Then you can go through, I would say start with the lined and labeled versions of these slides and go through them and test yourself. What type of epithelial tissue is it? So again, remind yourself, epithelial tissue is the general tissue type. Now let's go to the specific tissue type. Okay, this one is squamous epithelial tissue. Or here, this one is, again, or simple squamous epithelial tissue, okay? And this is simple squamous epithelial tissue, but it's frog skin. So I like this example of simple squamous epithelial tissue a little better because at least it's not frog skin. So it's just this outer serosa layer, these little flat, tiny pancake cells that are lining just the outside, okay? So, and again, you can kind of, here, you can see that a little better right here. This is the type of slide I would give you. And so, again, you can go through these slides and test yourself and make sure that you know where everything is. And then, if you really want to kind of test yourself, you can go to the lined versions and go, okay, let's see what I really know. Like, do I really know my stuff? Okay. So, okay, this is, simple squamous epithelium and kind of go through that and test yourself. And then lastly, you can go through and I have some soft chalk exercises, activities for you right here. So you can go through these and they're just great little labeling exercises. They're the drop and drag ones. And so I'm not going to go through those because you guys have seen those. And then here's the study mate flashcards. So hopefully all those tools will help you. If you want a little more, these are good videos, but he gives a little too much information. These are made for medical students. But if you want that little extra information, they are some great videos, so you can watch those. Okay. So again, I'm going to make a separate video here for connective tissue, and then I'll go ahead and send that out. But I'll go ahead and post this, and that's kind of what you're going to be doing for lab. Oh, last thing for the lab. So you can go back to return to the histology page. When you're done with all of that testing yourself, go take the practice quiz. Okay, and see, because it's a good indicator of kind of what types of questions and how I'm going to ask things on the practical. It also helps to remind you that you're not going to have a word list with you. So you really need to know these terms. And then as for spelling for the practical, and I know I'm going to get this question. So typically what we do for spelling is if you miss one letter, like let's say you left out a letter or something like that, we usually don't take off for that. If you kind of miss 2 to 3 letters, then, we usually give you partial credit. So I might take off 1 point for that. And then if you totally butcher it, but I can kind of tell what you meant, then I'll usually take a full point. Sorry, I take a half point off for the 2 to 3, and then a full point off if you've really kind of butchered it, and I'm not sure what you, I know what you meant, but you kind of really mess up the spelling. So that's kind of usually how I do the grading, and you'll get kind of a feel for it as we go through this first practical, but that's usually kind of what we do. So the moral of that story is do pay attention to your spelling and do try to spell these terms correctly. I mean, especially because you're going into the medical field. So you really want to know how to, spell these terms appropriately. So that's kind of the reason we do the practicals that way. And then we also were preparing you for nursing school and the boards and medical school or dental hygiene and all that. So we want to make sure that you're ready for all that. So anyway, I think that was all I had for you guys. That's my weekly announcements. I really apologize. This is way longer than I wanted it to be. But please, as always, if you have any questions, let me know. And I am more than happy to set up some time with you if you need some time as well. All right. Have a great week, all.